Okay.
So welcome everyone to here.
Hold on.
Okay.
Need a microphone from Zoom, yeah.
So this is it?
I'll let you know when
Wait.
Hello.
This.
Sure.
Sorry.
Alrighty. Thank you very much. Yeah.
I have
Sorry.
the signal.
No, it's fine for me. We just an introduction. Yeah. So hello everyone for coming for this week's NICO Wednesday talk. My name is Qing Jin, Associate Director of NICO. So this week we are very privileged to have Professor Satoru Suzuki. Professor has a background in physics at the very beginning, from master's degree in MIT, and also got his PhD in Harvard University about visual perception. So he has done a lot of brilliant works about the visual perception and the psycho-physical relationships, and also uses some like computational mathematical models to understand this system. So today's talk is about some simple rules governing microscopic oscillatory neural activity in humans. Please join me in welcoming Professor.
Oh, well, I don't need that. Yeah. Thank you very much, Professor, for the invitation. I'm very appreciative and honored to be here. And also, I'm very happy that so many of you came to hear my talk. Thank you very much. So let's get started. There's some preview of the data to be presented, so you can look forward to that. So my talk is going to be about oscillatory neural activity, which is ubiquitous in the brain. And the reason is that many cortical pyramidal neurons, the cortical pyramidal neurons are the dominant excitatory neurons, which comprises about 80% of excitatory neurons, also whose activities are detected by surface recorded EEG. So it's very relevant to my research. So those pyramidal neurons, cortical pyramidal neurons, are connected to local inhibitory neurons with specific onset and decay rates, thus generating oscillatory activity at specific frequencies. So I put a little diagram. So the P is the pyramidal excitatory pyramidal cell, and they tend to get excited, but then that triggers the associated inhibitory network. That's going to kick in with some onset delay, and it brings down the excitability, but then the inhibitory influence decays, going back to being excitable, create a sort of oscillatory cycle. And then you can intuit that if the onset delay from the inhibitory input and the decay rate are faster, then that would create faster oscillation. Whereas if the onset and decay rates are slower, then generates a slower oscillation. So, a lot of pyramidal cells have oscillatory activity of different frequencies, depending on the rates of the onset and the decay rates of the associated inhibitory network. Okay, why study oscillatory neural activity? I can't really get into the gory details, but oscillatory neural activity enables flexible control of neural communication and also does many more things, primarily by adjusting within frequency phase alignment or using slow oscillations to temporarily organize faster oscillations. So if you want to know about more about these functions of oscillatory neural activity, these are excellent reviews that you might want to look at. So for now, I'd like to just illustrate one, I think, important function of oscillatory neural activity in terms of controlling neural communication in a very diagrammatic way. So let's look at this. So this is neuron A, B, and pyramidal neuron A, B, and C. They are physically connected. And suppose that they are engaged in oscillatory neural activity. So the main brain potential is oscillating between excitable, less excitable, excitable, less excitable, plus to minus. And then the action potentials are likely to be generated during the excitatory phase, indicated by these vertical marks. And in this particular case, because the oscillations of the three neurons are aligned, the excitatory, sort of the action potentials generated here would be transmitted to exciting neuron B. Also, action potentials generated in C will be communicated to neuron B, and then they both will be likely to successfully activate neuron B because they arrive at the point where the B is excitable because of the in-phase alignment of the oscillation, okay, schematically. However, if you change the phase of neuron C to be out of phase, then despite the fact that the physical connections are present between A and B and C, you can restrict the communication between just A and B just by shifting the phase, because these action potential signals would be arriving when B is not excitable, so unlikely to communicate to B, but A will be communicating to B. Going back, now there's a communication, equal communication from A to B and C to B, but then if you misalign A with respect to B, then you would be promoting communication between C to B and kind of blocking the communication between A and B. So in a way, even though the physical connections between neurons in the brain change very slowly through learning, but we can have different thoughts that are changing rapidly. And then those sort of cognitive processes that require a variety of different communications happening, changing all the time, it's thought to be mediated by this sort of a mechanism. So changing phase to sort of selectively increase certain communications and then disabling other communications. Okay, investigating oscillatory neuroactivity. Most studies, if you look into the literature, studying oscillatory neuroactivity, aim to relate oscillatory activities in specific frequency bands like 10 Hertz alpha or 15 Hertz low beta and so on, in specific frequency bands and brain regions such as in frontal areas, prefrontal cortex, parietal lobe, to performing specific cognitive functions such as attentional selection, working memory, episodic memory, decision-making, motor control, and so on. And then there are tons of interesting results regarding the neural correlates of sort of major functions that we, cognitive functions that we perform each day in terms of what sort of oscillatory activities are related to those neural correlates. There are tons of interesting results. So we tend to, well, at least I tend to get attracted to things that other people are not doing. So in contrast of this great body of literature, the goal of our recent curiosity-driven research has been to discover simple rules that govern the global dynamics of macroscopic because we're not using microelectrodes, we're using a scalp EEG electrode, so we can only detect macroscopic the neural activity generated by large populations of neurons, oscillatory neural activity. So simple rules that we're interested in. Why? We need to justify a little bit. We can't just say we're curious. So one thing is, okay, simple macroscopic rules may provide useful constraints on neurocomputational models of global neural dynamics. I'm sure that in this audience, many of you are studying neurocomputational theories, models of neural activity. But for us, more important reason is that discovering simple rules obeyed by a complex system with about 86 billion neurons is just simply very exciting. It's a complex system obeying very simple rules is exciting, and it's discovering those simple rules is by itself, like regardless of whether it's going to be applicable to anything, it's very exciting to us. Okay, now we're going to switch to general method before presenting you with the data. So what we do in general is that we need to extract oscillatory neural activity from the EEG signals. And then we do so by running wavelet templates. As you can see, these wavelet templates, which are tuned to different frequencies like lower frequency, medium frequency, high frequency. We run those wavelets through the raw EEG data and then compute the dot product, which is basically you multiply the EEG and the template at each time point, which is can be thought of as a computing correlation at each time point. So what you can intuit is that if there's an oscillation of this particular frequency present in the background EEG, the dot product at that time point or correlation would be high. So what that means is that if you run these wavelets through EEG, where the correlation is high indicates that there is a substantive oscillation of that frequency. Okay, so that's how we detect oscillation using these wavelets. Also, you notice that each of these wavelets have a cosine component in red and a sine component in blue. And if you do this dot product and assign the cosine component of the dot product to a real part, and then sine component to the imaginary part, I don't want to get into the real details, but you can look at the sort of a symbolic equation there. You can extract both the amount of oscillation at each time point, how strong the oscillation is, in terms of the amplitude squared, which we call that power, and the phase. Phase is what part of the oscillation cycle that particular oscillation is at the top, bottom, back to the top again, like it could be 0 degrees, 90 degrees, 180 degrees, 270 degrees, and 360 degrees. Okay, we can get that information. So in essence, what we do is using these wavelet templates of the tuned to different frequencies to extract the strength of oscillation and the phase of the oscillation at each time point simultaneously. That's what we do. Okay, so that's the basic method. Now, because we're interested in general rules that governs dynamic oscillatory activity, our subjects, research participants, I guess, are engaged in sort of a natural thing that they do. They close their eyes and then think about whatever they want to think about, or they view a nature video that's very relaxing. And then while they do that for about 5 minutes, we record the EEG. Okay, so that's a part of the general method. That's a scalp map, and then we have 64 electrodes, and we surface Laplacian transform EEG in order to localize the sources on the Dura surface. I can get into the literature, but right now I just say that much. And if you do that, you can localize images. The Dura surface is basically if you open up the skull, and that's the superficial, most visible surface of the brain. And we have shown, and others have shown, that if you do that, you can get the spatial resolution of about 1 to 3 centimeters, which for the 64 electrode array we used corresponds to about the average spacing of the electrodes, indicated by the dotted circle. So that's our spatial resolution of our EEG data. Okay, another thing to think about before I go into explaining our results, in EEG, larger oscillatory power, which we call spectral power, or SP, I'm going to be using that notation later on, to indicate the oscillatory amplitude at a specific frequency. So oscillatory power indicates synchronization, the amount of synchronization of a larger population of neurons. So let me just illustrate with a diagram. So if a bunch of neurons, it's not going to be just 4, it would be thousands of neurons under each electrode. If they're all synchronized, they all get summed. So you can get very large oscillatory EEG amplitude or power, right? But even if these oscillations are going on at the individual neuron level, if they're not synchronized, then there's going to be a lot of cancellation. So the EEG is going to be very small in terms of the power. Okay, so in EEG, unlike signal cell recording, the large oscillatory power means that a whole bunch of larger number of neurons are synchronized, whereas a lower oscillatory power means that the fewer neurons are being synchronized, or so most neurons are desynchronized. So that's going to be the interpretation because we're using EEG as opposed to signal cell recording. So if you have 2 populations, the question is, what if there are 2 populations underneath the, I guess, the electrode that were in antiphase, then we would get nothing because they would cancel. So that's the limitation of using a macroscopic method like EEG. We can't distinguish between canceling versus absence. Yeah, very good point. Okay, so this is an example of oscillatory power, which indicates the amount of synchronization that we computed using these wavelet templates spanning about 3 Hertz to 60 Hertz. And then these colored lines indicate the data from these representative locations on the scalp. And then what I want you to show is how, this is one participant, like, sort of contemplating things with the eyes closed, how the oscillatory activity is changing over time. This is real time. If you want to know how the time is progressing, it's indicated here in milliseconds. this is 8 millisecond point, and it's going to increase to about 4 seconds at the end of this movie. So I just want you to look at it. So this is somebody's brain sort of having random thoughts, and the oscillatory activity in different frequencies are just changing. I'm sure they underlie the changes in the thoughts and stuff like that. It's seemingly random, or maybe not. It's very interesting, and you can look at these things in conjunction with performing a specific task, and then understand 30 hertz oscillation in the middle part of the brain corresponds to working memory, something like that. You can make that kind of inference, but what we are interested in is, look at this. Are there any simple rules that this seemingly random behavior obeys? So that was our sort of curiosity throughout the topics that I'm going to be presenting in this talk. So it's a few more seconds, just enjoy this movie. Yeah, so that's how I got captured into this. Like, I made a movie a long time ago, and I really wanted to understand, like, what's going on? Are there any simple rules? Because I'm interested in some things that are simple, as opposed to things that you can only detect with machine learning. I'm just interested in simple rules, at least for now. Okay, so the roadmap is going to be, I will talk about 3 simple rules that we studied, but for each of them, I'm going to talk about our initial curiosity. What were we curious about, in terms of deciding to investigate a particular phenomenon. And also we talk about the means by which we derived, each of these simple rules. So that's going to be the roadmap. So we're going to be talking about 3 things, which are loosely connected, but you can also, in your own mind, you can connect them. Maybe you can have better insights than me, because I've been spending so much time with this. So I may need some fresh insights from you guys. So the first curiosity we had was this: are there any global rules that are simple governing how maximal and minimal spectral powers corresponding to maximal synchronizations and minimal synchronizations occur at different sites? So if you observe, the movie like that, sometimes here there's going to huge amount of synchronization, maximal synchronization, and it goes back down. And here there's going to be huge synchronization. Sometimes in 2 places you can get huge synchronization, maximal synchronization, and sometimes you get sort of quite suppressed amount of synchronization or desynchronization at this point, or multiple points at the same time. But the question is it random, or are there any rules that govern, that kind of occurrences of maximal and minimal synchronization? It's kind of interesting because maximum synchronization, or the maximum power in EG, means that whatever the thousands of neurons underneath that electrode is maximally synchronized. It's got to mean something, right? When you look at extreme cases, maximal, minimal, it's highly unlikely that it's just an epiphenomenon. It's got to mean something. So we were, for that reason, extra interested. Okay, so now we're going to go directly into the data. But before I'm going to explain all of these things, so you can read them, I want you to just look at it and then notice something, and then see if you agree with me. So when I look at this graph, it's a time series of something, right? Data. What I noticed from this is that the brain tends to spend a lot of time in 2 different states, and then jump between sort of 2 different states. One state is low, being low, or being high, or being low, or being high. But in the middle, it's transitional. It just goes zoom up, zoom down. So it seems that there are 2 different states that our brain, or this is just one person, this person's brain is going, switching between, like a low state and high state. Do you sort of see that in the data? Okay, so then I'm going to sort of explicate it a little bit. So the y-axis here is the number of sites that are maximally synchronized. So low number means that here, if it's 2, then only 2 sites had maximal synchronization. It could be these 2 or these 2. I don't really know at this point. But the low state means that only a few sites are maximally synchronized. The high state means that in this particular case, 15 sites were simultaneously maximally synchronized, something like that. is happening. So what this data is suggesting is that the maximal and synchronization happens in two networks, small scale network in this case, and a large scale network in this case, and then they kind of hop between them, the two states. Okay, so that's what I kind of get from this diagram. So this is one subject. So you have to actually do a statistical sort of analysis. So what's presented here was computed the amount of time is spent in high state. So this is amount of time here, amount of time here, amount of time here, and then we average them, and then how long it's spent in high region per instance, and also low region can be the amount of time here, amount of time here, amount of time here, and then average, and then compare that with a control, which we generate by phase scrambling the data. So phase scrambling, what it does is that you preserve the average oscillatory power of the EEG, but you just randomize the order. So that's a good control, and I can talk more about it if you're curious at the end. So you can see there that the compared to the control, PS, the duration of staying in the low state or small network state is higher. And then dotted lines indicate the individual subjects. So it's a very strong data. Almost all subjects out of 24 show that trend. And also the time spent in high state is also much higher than the phase scramble control. And then all subjects show that. Whereas in the middle state, in the transitional state, is comparable to random data. So it supports this observation that our brain tends to stay in a low state like this and high state like that, and then jump between the two. And we found something similar, not with just the maximal synchronization, but also minimal synchronization showed the same kind of pattern. So this is the minimal synchronization pattern, otherwise the same format. You can see that again, the brain tends to spend in the low state or high state, but the middle state tends to be more transitional. So low state meaning that only 2 regions or 3 regions had a minimal synchronization, but the others were doing other things. And then in the high state, then a whole bunch of sites, brain regions, were engaged in minimal synchronization. Again, it seems to switch between the two. And then we did the same kind of analysis like we did there. You see the similar sort of thing. Compared to the phase scramble control, the duration of a small scale network state is increased. Large scale network state is also increased relative to control. But the amount of time in the transitional state is the same as expected by chance or random sequence. So now the question was, okay, there's this, the brain tends to be in a state where small network of high activity or large network of high activity, but were those networks like randomly chosen from the brain, or were there any spatial consistency? In other words, is it the case that these networks always occurred in certain region, and then these networks occurred in other regions? And if so, what is the relationship? Or maybe no relationship. So that's what we looked at. So what we found, so this is a topographic map of the instances occurrence of it's a low state. So number of sites, only one site being maximally sort of synchronized, happen in these regions. So it's relative to a chance. So dark blue indicates below chance, and the dark red indicates above chance. And then colors that are corresponding to the top and bottom is a statistical significance with 48 participants. So what you notice is that it is localized. So isolated maximal power activity, not shared by other regions, tended to occur, this is for alpha, by the way, in frontal regions selectively, like this. What about like a more spread out high synchronization activity? We found it in sort of those regions. So these are corresponding to 10 regions simultaneously being maximal spectral power, 11, 12, 13, and so on. You look at this, you also notice that there's a pattern. This pattern, small network activity, and then large network activity are complementary. It looks like they're complementary, right? So to test that statistically, we computed the cosine similarity between the first one and the second one, first one and the third one, the first and fourth one, the first one and the fifth one, and so on. So this is the first one and the second one, fifth one, and so on. And then cosine similarity goes from below 90 degrees is similar to this. 90 degrees is orthogonal. And then closer to 180 degrees is opposite. And the gray lines indicate individual subjects. The black curve is the average. So you can clearly see that for all subjects, consistently, the pattern went from one pattern for the small scale network, and then it switched to the opposite pattern in the large scale network. If you're wondering about the I'm not supposed to move out of this bar, I'm sorry. If you're wondering about the open circles, that is the cosine similarity computed not per subject, but they computed on the average. Literally was computed on these patterns, the average patterns, then the effect is stronger. What that means is that by averaging across subjects, you can get rid of a lot of noise. So that's why the effect is stronger when you look at it on the average data as opposed to looking at it on the individual subjects. We found something similar for the minimal spectral power or the maximal desynchronization. That's for the small scale network and the large scale network. So what's very interesting to us is that the same network around here engages in both like sporadic maximal activity and a sporadic minimal activity. So they are sort of uniquely engaged in sporadic activity, whether it's maximal or minimal. And also in these regions, somewhat similar to there, this particular large network is engaged in widespread high activity or widespread low activity in terms of synchronization, and they're at complementary locations. And so this is the same plot, cosine similarity, so the same as above. It shows that these patterns are complementary spatially. Okay, so one caveat or additional point is that this is alpha frequency, which is about 10 hertz. So this complementary topography between the small scale maximal minimal activity and the large scale minimal maximal activity were true for all frequencies that we examined, but the exact topographic patterns were dependent on which frequency we're looking at and which condition we were looking at, whether you have your eyes open, watching a movie, or eyes closed. But the basic rule that we are proposing here of this complementarity between the small scale maximal minimal activity and the large scale maximal minimal activity was common in all conditions and all frequencies. So therefore, we claimed our first rule, simple rule. I think it's simple, although there's a lot of data. So the maximal and minimal spectral powers, which indicates maximal and minimal synchronizations, occur in spatially complementary small scale and large scale networks in general, okay, across different frequencies and conditions. Okay, so now the second topic. So now we switch gears a little bit. We're interested in phase relations now. So previously we looked at power, like the strength of oscillation, but now we're interested in phase relations. So before, so we're curious about, are there any rules governing global phase relations between different pairs of regions, I guess. So that's what we're interested in. Are there any rules? Because we know that the phase relations change depending on performing different tasks. There are tons of papers on, like, for instance, where you engaged in working memory, the phase relations in alpha change in certain brain regions, stuff like that. But we were interested in sort of a general rule, like, which would be true no matter what you're doing, that may govern the global sort of interaction of phase relations. To get into this, I'm going to sort of refresh maybe your memory a little bit about phase relations. So phase relations in EEG, like this, we pick two regions, two sites, right? And the delta theta is the phase difference. So we measure phase relations in terms of phase difference, which is delta theta, site 1, site 2. So this would be an illustration of a near synchronization. So if the oscillation in site 1 and site 2 are kind of like this, so delta theta would be near 0, and you can argue, see that it's synchronized basically. Okay, so if delta theta, the phase difference was pi/4 or 45 degrees, then it's kind of like this, meaning that site 1 is leading site 2, somewhat synchronized, but there's a delay. Okay, 90 degrees, and then as we pointed out, 180 degrees would be antiphase or out of phase, meaning that one side is in upstate and the other one is in downstate. they would be summed to 0, for instance, if they were coming from the same location. So in this part of the talk, I'm going to be talking about so-called phase flipping. So whenever I say phase flipping, what I refer to is the relationship, a phase relationship between the two sites flipping from near synchronization to near antiphase. So this is what I mean by phase flipping, and that's going to be happening. That's going to be sort of a major component of the next part. You guys all right? Okay, so if you want to think about phase relations, global phase relations, are there any simple rules? It's so vague. So you have to have some kind of a hunch as to what sort of rules may be applying. So what we focused on was modularity, okay? Instead of reading that, I'm going to give you an example. I think the example makes it sort of clear sometimes than the description. So we can measure modularity, which I can talk about it later, with this quantity Q. So high Q means that it's a highly modular state, and then low Q means low modularity state. So this part corresponds to the phase relations across 64 sites and 64 sites. Each point indicates a pair in the top 5th percentile of Q value, and here is the bottom 5% of the Q value. So what you notice is that, okay, when the modularity based on phase difference, absolute phase difference is high, what you see, this is one participant's data, but this is representative. It's not the best, it's representative. You can see these clusters. So this participant has 3 clusters. Within the cluster of electrodes here, or the sites here, they're highly synchronized. Okay, so high synchronization, low phase difference within cluster, but between clusters, there is a pretty high phase difference, meaning that within cluster, the sites are synchronized in oscillation, but individual clusters are not synchronized with one another. So when we talked about phase relations, there's a good communication when they're synchronized, but sort of a blocked communication where they're desynchronized. If you think about sort of in that way, you can imagine that this is a state where these clusters of sites, brain regions, are highly communicative at doing their own things, but they're sort of independent of one another to some degree. And the low modularity state is like this, like all of these, it's not clustered like this, but all of the sites are somewhat phase related. So the values of the phase relations. So this is a histogram of all the values of phase differences, absolute phase differences. You can see that it's mostly somewhat synchronized, and then the cases of a desynchronization is quite low. But in the modular state, what's really salient is that a lot of pairs become anti-synchronized here, or out of phase. So that's a big difference here. So what we see is that your brain right now, because this is like we recorded this kind of data when people just close their eyes and thinking about things or watching a nature movie, in your brain right now, this is happening. Your brain is oscillating between this kind of state, this kind of state, this kind of state, and this kind of state. So what we were interested in was, okay, what kind of networks controlling the switch between this and this and back to this and back to this? So that's what we're interested in. So in order to find out, because we know that the difference between the two states is the increase in the antiphase relations, we wanted to find out, is there any specific network that flips between antiphase and in phase? Because in the case of a low modularity state, these pairs would move to here, and in a modular state, those pairs would move to there. We wanted to see if there's a specific network that was doing this, as opposed to different brain regions are doing this depending on whatever is going on, because we were interested in sort of simple and consistent rules. Okay, to look at that, we plotted each phase pair in terms of what the phase difference is in the high modularity state on the y-axis, and then what the phase difference was in the low modularity state. So each point represents the specific phase pair in the high modularity state and low modularity state. Specific phase pair meaning between a specific pair of electrodes or the sites. What we noticed is that they clustered in 3 regions. So in the boxed regions can account for about more than 70% of the total pairs. So we focused on them. So this here is sort of the pairs that stayed in low phase angle throughout, regardless of condition, or did not fluctuating, because, for instance, this pair would have a very low phase difference in the high modularity state and also in the low modularity state, right? And then that one would be the opposite, stayed in the high phase difference, almost antiphase, regardless of whether the state was in a modular state or low modularity state, high or low modularity state. The most interesting group was the upper left corner, because they actually flipped. They had a high phase relation in a modular state like this, but they flipped to low low angle difference in the low modularity state. So we were really interested in those. So to confirm that these switches were, actually going on, we plot a little bit more detail. So this one is comparing the high modularity state and low modularity state, meaning top 5% in Q versus bottom 5% in Q. In this graph, I'm plotting the histogram of the phase difference for all of the Q values, going from 0th percentile to 95th percentile. Yeah, question.
This graph, that a single subject? A single subject.
Oh, very good point. So this is so all of these dots are time averaged phase difference, but averaged only for the high modularity state. So there are high modularity state in this time segment, this time segment, this time segment. I average the phase difference across those segments. So these are time averaged. So each point is one phase electrode pair from one participant. And then all these dots, actually, I should have mentioned this, include 21 participants. So that's why there's so many points there. Very good question. Any other questions? Okay, so indeed, for these pairs, electrode pairs or side pairs, the phase relations always stayed low, regardless of the changes in Q. Always low phase relation, angle differences. And for those, always stayed high, regardless of, so meaning that their phase relations were very stable over time, regardless of the changes in degree of modularity. So the most interesting one there is the upper left. Indeed, they flipped. They had a low phase angle difference in the low modularity state, going from 0 to, I don't know, about 40th percentile, but then flipped to a high angular difference in the higher modularity state. So they really drive this change in modularity by flipping their phases. So then our question is that a specific network, or is it all over the brain? So that was our question. So what we did was very simple. So we took all of these, and then we sort of plotted where they exist. And it turned out that this is a consistently sort of synchronized type of relations tended to exist between sort of connecting homologous locations across 2 hemispheres. So it's kind of interesting on its own, and there's some differences based on frequencies, but we weren't focused on that, but I'm just showing you because it's interesting in its own right. And also these pairs tended to consistently across subjects show high sort of antiphase relations. By the way, if you do a statistical analysis, these tend to be horizontal more biased, and these tend to be sort of vertical biased. But the most interesting was, okay, is there a consistent network that flips the phase between low and high, which would be driving the modularity from high modularity to low modularity to high modularity? What we found was very interesting. So regardless of the frequency that we're looking at, we found the anterior-posterior network. That is the thing that's flipping phase between low phase and then high phase, low angle and high angle, therefore driving the difference between modular and then less modular state. Always like the network that is, frontal and then occipital or parietal long distance sort of relationships. And then we found that although the network related locations are similar, or pretty much the same across statistically identical for different frequencies, the switches were not correlated, meaning that when this frequency, there's a switch, doesn't mean that there's a switch in 15 hertz. So what that means is that the modularity, phase modularity, is controlled separately for different frequency bands. So even if it's happening here for alpha range, beta range could be like this. Similarly, when the gamma range is like this, theta could be like this. So what this suggests is that the modularity, whatever that is meaningful for our functions, is controlled separately for each frequency band. And okay, so to summarize, the simple rule that we derived here is that a phase flipping anterior-posterior network indicated there controls global phase modularity. It's quite simple. Okay, so I have some time left, so I don't have to skip the last one. Okay, so the last one, I think, is the most recent thing that we have been doing. So we're basically excited about it, but a little bit more nuanced. So the question of curiosity is, how are oscillation frequencies, which is spectral peaks, I'm going to show you in a second, driven to converge? So oscillation frequencies can be happening in different frequencies, but in different regions. But when they converge, meaning that if they equalize their frequencies, that really facilitate interactions. So equalizing frequency is kind of an important thing. And we're interested in what kind of mechanism may be driving that equalizing of frequency. So before getting further into it, I'm going to show you a movie. So this is sort of a broad alpha range going from 5 to 15 hertz. Maybe this could be theta and this could be low beta, but broadly speaking, it's alpha range. And I'm going to show you spectral power, how it's changing. And these peak locations indicate dominant oscillatory activity going on. So this is somebody's alpha with eyes closed. So, and then these different curves represent different sites, which I'm not going to talk about it right now, but all you need to think about right now is that I want you to observe that sometimes these oscillatory peaks like dynamically converge and then disperse and converge. And I want you to see if you notice any kind of regularity to it, any kind of rules that you might be able to intuit, because that's what the, well, that's not what I'm going to talk about. It's a mechanism. But anyway, so that would be good. And also, this movie is going to be almost real time. So the time is showing in seconds. And if you keep track of the time, it goes 105, 106, almost like 1 second at a time. So this is real time, somebody's brain, how the oscillation is changing, converging and diverging. I find it to be quite interesting. So this is alpha range. So you can see like a convergence of different sites sometimes, and then they disperse and, higher power. You can see like convergence like moving together sometimes. Right, sort of, they go up and they kind of move together. That's sort of, that was a very strong convergence there. So again, there was a convergence there. There's going to be maybe 10 more seconds of this that you can enjoy. I just find this kind of movie to be just so remarkable. This is somebody's brain in real time, oscillatory activity. And it's all there for us to understand what's going on. How these things are happening. It would be nice if there's a model to, predict exactly how these things evolve. I'm sure some of you guys are doing it. So this is the end. And then you can also see that there's lower, upper alpha and lower alpha like happening simultaneously and stuff like that. You can see all of that. So in this part, I'm going to be focusing on the anterior-posterior relations, frequency convergence occurring across the anterior-posterior axis. The reason is that recent, very interesting studies show very interesting global gradient along the anterior-posterior axis. You can read these papers if you're interested. For instance, the myelination decreases from posterior to anterior, meaning that the neural conduction speed is decreased going from posterior to anterior systematically, and also cortical thickness connectivity, amount of connectivity, and the amount of temporal integration of neural processing systematically increase as you go from posterior to anterior. So because of this sort of general gradient that has been discovered recently, we focused on the frequency convergence along that axis because frequency convergence facilitates communication, and then there's interesting gradients, so that may be more relevant. Also, in this particular study, we focused on the alpha range because alpha range, probably it's like a very well-known frequency band implicated in a variety of perceptual, attentional, and memory processes, and also known to influence gamma band activity, and also known to temporally organize visual information. So we focus on the alpha range. Okay, so I'm going to show you sort of a graphic version of this movie, so to speak, here. So what this shows is, this is going to be the last slide you're going to see before the summary one. The summary one is kind of not taxing at all. So here's a frequency here. And then these different colors indicate how many sites along the anterior posterior axis were converged in terms of their peaks. So the dark blue indicated by the arrows means that at this time point, at frequency 8.5 hertz, all 8 sites were converged to 8.5 sites. What that means is that in all 8 sites, there was a peak at the 8.5 hertz at this time point, and so on. So dark blue indicates greater amount of convergence, and then other colors indicate intermediate to low levels of convergence at different frequencies. So we wanted to understand investigate was what mechanism is driving this convergence? Is it that some brain region driving this, ordering these anterior posterior region to converge, or this is more like happening spontaneously and in a certain way? So again, this is a vague question, so we had to have a hunch. So a hunch was that it's not like there's a driver. There could be microscopically, but macroscopically, we thought that the emergence of these convergences would be synergistic, like sort of as a spontaneously arising sort of organization. And if that's true, we hypothesized that the probability of increasing in convergence, going from 2 sites converged to 3 sites converged to 4 sites converged, the stepping up in convergence would increase the probability as a function of the size of convergence. So if there's a synergistic interaction, going from 2 to 3 would be harder, but the 3 to 4 would be easier, 4 to 5 would be easier. So that would be like a synergistic interaction. So we looked at these transition probabilities, going from -1 to, well, k minus 1 to k. And indeed, we found that. So the probability of kth convergence occurring as a conditional probability given k minus 1 convergence, what is the probability of kth convergence occurring, is increasing as a function of the size of convergence, as I pointed out. So these ones are control conditions. So this is an important control condition where we apply the same analysis while we randomly shuffle the time of the time series of peaks for different electrodes. So that preserves all of the pattern of where you find peaks, likely to find peaks, but just shuffle the destroy the temporal dependence. So that this is a very good control. But even with respect to this control, there was a systematic increase in the probability of convergence as a function of convergence. So this is an indicator of synergistic interaction. So given that, we were interested in, okay, it's maybe synergistic interaction. So what is kind of underlying macroscopically this synergistic interaction? So to give us a hint on that, we found this relationship. So what we looked at was the oscillatory power that we talked about as a function of convergence. So these are color-coded different sites here. So for all sites, I mean, this is a little bit weaker, but for all sites, the power amount of synchronization occurring at each site systematically increased as a function of the convergence of frequencies to which they were participating. So what this suggests, not means, but suggests, in combination with synergistic interaction that we just sort of showed, is that what's going on maybe is when frequency convergence happens, like between, say, 2 sites, that boosts the amount of oscillation because of the synergistic activity. So the power increases for, amount of synchronization increases for here and here by virtue of convergence. An increased amount of synchronization may facilitate the entrainment of neighboring regions. So it's going to spread, synchronization even more. So we feel that these results combined may suggest that the synergistic interaction may be driven by boosting of oscillatory synchronization by virtue of the strong interactions because of the alignment of the oscillation frequencies. So the last thing I'm going to talk about in relation to this is, okay, alignment of synchronization that may facilitate communication across the anterior posterior brain region activity, but is there any sort of hint as to what sort of thing that we may gain by the brain is gaining by aligning? So we looked at phase relations. So we looked at phase relations from posterior to anterior. We arbitrarily set the posterior phase to be 0, just that you have to set a baseline somewhere. And then these are phase differences, average phase, time averaged phase differences going from posterior to anterior. And then we looked at these phase differences in 3 different anterior posterior strips, in the middle, in the left hemisphere, and in the right hemisphere. Interestingly, we got this sort of, we didn't expect this, surprising spatial segregation, meaning that the phase gradient is negative going from posterior to anterior in the middle. What this means is that the phase is, delayed further, you go further in anterior. So what that means is that posterior is going up, and the next one is going up and up a little bit later. So this sort of phase gradient may suggest that the oscillatory information is flowing from posterior to anterior in the sense that posterior is leading, goes up, and then this goes up, goes down, goes down, goes up, goes up, down, goes down. So in a simple sort of correlation kind of idea, you can imagine that this is driving this one because going like this in this kind of gradient. And then this is the opposite, because in this case, the interior, more interior is leading more posterior. So it's the opposite, but this is leading and then goes up, going down, going down. So, one may conjecture that the information flow is in the opposite direction. The other thing you notice from this graph is that the steepness of the gradient, phase gradient, increases with the size of convergence. So what the convergence is doing, alignment is doing, is to make these information flows, if you think of it as information flow, more strongly directional. Why am I saying this? Because all of these phase relations have variability, okay? But we found that the variability was the same regardless of the amount of convergence. So given that random variability of these values are the same, if you have steeper gradient, that means that the gradient is more reliably different than 0, that is. So we think that the, one of the things that the frequency convergence does is to enhance the directional information flow between anterior and posterior sort of areas. And the last thing I want to talk about in this, or not second last thing, this very last thing is not that important. So this spatial segregation, interestingly, was specific to eyes being closed. So right now you have your eyes open, you don't have this, but as soon as you close your eyes, you have this. So when you close your eyes, you kind of really spatially segregate the information flow so that it's from posterior to anterior, in the middle, and then in each of your hemispheres, the information flows kind of like this and going back like this. When you open your eyes, this kind of disappears. So to me, it's kind of very interesting that, if I think about it that way. Okay, you have a question? So she has a very interesting question. Is the emergence of this pattern, is it specific to closing eyes or absence of visual information? That's an extremely interesting question. That's a question. So we did have those conditions, actually. So because of the time, but I still have like a few minutes, so I can just get into it a little bit. So what we found is the answer is kind of partial. So this pattern, this segregation from a individual subject, this is the average across subjects. Individual subject base is very strong when they have their eyes closed. We replicated this many times, even when people were doing different tasks. So we had people close their eyes and then do a working memory task or a vigilance task or mental calculation, you get this. So this doesn't really depend on what people are doing, but it's just by closing eyes. Now, in another condition, we had people open their eyes, but put them in a really darkened room. In that case, it was intermediate. This segregation got a little bit weaker, but not completely eliminated. So that's always a sort of disappointing result in between. Eyes open is you don't get this segregation. Eyes closed, you get clear segregation. If you do something in between, like eyes open with darkness, we got something in between. Great. So the last thing I want to point out is not that, it's just to show that as you can see, the strong frequency convergence for this particular subject is happening in the alpha range, although we looked at, from theta to low beta, but actually the strong convergence is, statistically speaking, happening in alpha range. And then we confirmed that for all subjects. So this is the average amount of convergence, and it is indeed focused in the alpha range for different strips of anterior posterior electrodes. Okay. So the final rule that I wanted to point out is frequency convergences along the anterior posterior axis in the alpha band are driven by synergistic interactions and enhancing phase gradients. All right, so we're pretty much done. So the goal was to show some simple rules governing macroscopic oscillatory neural activity in humans. We talked about 3 rules. The rule number 1 was that maximal and minimal powers or synchronizations occur in spatially complementary small scale and large scale networks. So the maximum minimal activity jump between small scale and large scale in the complementary brain regions. Second rule we found was that a phase flipping anterior posterior network indicated with those red lines network controls global phase modularity, in that when these relations are high angle, the brain gets into a highly modular state. When the phase relations in this network is low angle, the brain gets into a low modularity state. So that network basically controls your brain state in terms of phase relations between highly modular and then highly non-modular. And then third rule we just talked about, the frequency convergences along the anterior posterior axis in the alpha range are driven by synergistic interactions, enhancing phase gradients. 2 remarks. Okay, these rules may provide macroscopic constraints on computational models. And if I were to say write a grant, I would also have to say, I'm interested in how these rules actually contribute to cognition and the maintenance of adaptive, responsive, or metastable neural dynamics. So these would be, the questions that we would be pursuing in the future. And thank you. And I also thank our collaborators, Melissa Mensoglu and Marcia Grabwerki. Thank you very much for sticking around. I know I presented a lot of data. Thank you, appreciate it.
Okay, so you show the probability of K given K minus 1 to increase up to 8. What happens at 9, or do you never get to 9? Because I think you were saying there was what, like 64 different sites?
Oh, very interesting question. So maybe I didn't make it clear. So in this particular analysis, right, we only looked at the 8 anterior posterior sites. So the maximum is 8 in this particular case. But in our more recent research, because we wanted to get something sort of tractable in terms of analysis and presentation, but we are looking at all 64 right now in terms of how, but if you have all 64, it's very tricky to, because also you can't really plot phase relations in this linear fashion if you have lateral kind of array instead of just a linear array. So we focus on the linear array here, but yes, we are now looking at convergences that's happening across the entire, say, brain, so to speak. But in this particular case, it's maximum is 8 only because we only looked at 8.
Okay, so it does increase over the entire set. I wondered if it dropped off at some point. If there's a limit to how many of these can converge, I guess.
Right. Yeah, there is. Yes, that's right. That's right. So we looked at, but I haven't actually tabulated data in terms of, okay, when you look at all 64, for instance, right? At each instance, you can plot how many, just like a, not kind of graph like this, like how many are converged, right? As a function of time, you can plot that. And off the top of my head, it varies, if I remember correctly. If you look at all of them, because there's a lot of chances, so the chance is higher too, varies from, I think, teens, around like 10, 12 is kind of common low end, because by chance you can have convergence like that. And for most subjects, in most cases, up to maybe 40 out of 64. But for some subjects, in the brief moments, can go up to even 50. That's my recollection. Yeah. Oh.
Understand that the oscillations are determined by the electric fields, but you could also have a spiking happening at the same time. Can you comment on any relation between the neurons firing versus the oscillations that you are discussing?
Yeah, so this very good question. So the problem with EEG is that, I didn't do it, try to figure out the relationship between spiking activity and the scalp recorded EEG myself, but based on my reading, EEG is pretty much dependent on the, local field potentials that is averaged across thousands of neurons underneath each electrode. So can't really distinguish between more spiking activity or less spiking activity per se. That's an unfortunate thing about, having electrodes on the scalp.
Thanks.
On this last question that you asked about
Yeah.
the frequency convergence, do you have any, to me, the natural complementary question is, why does the synchrony break apart, or why does it fall apart, and how long does it last? Do you have any thoughts on what determines that timescale for how long?
Oh, so how long? You can kind of get it from here, right? Because this is in seconds. Although we computed statistics on that, but I don't remember off the top of my head. But this is a representative data. So this would be like 8. In fact, in the paper, we present that data, but I don't have the paper right now.
You can see how long it lasts, but do you have any
Right.
theoretical reason to explain why?
Why this is the duration that they are lasting? It's based on some sort of computational model, you're saying? The short answer is no. That would be interesting to know. That would, get into the realm of like you study like coupled oscillators and then manipulate different parameters, coupling strength, or that is the symmetric part or asymmetric part, and yeah, but we haven't done anything like that. Yeah, but it's a very good question. Thank you.